So in this presentation, I will present you the main codes in our portfolio and tell a bit more about how they can be used in a scalable and efficient way on HPC infrastructure. For those of you who are not familiar with BioExcel, just very briefly, it was established in 2015 with three main objectives. One is to provide life science researchers with high quality and user-friendly software. This is imperative. We need that, and it takes a lot of effort to make it high quality and user-friendly. But both of these require a lot of work. Not only that, we don't do only software engineering, but also work on increasing the expertise and the skills of researchers with a very extensive training program, which I will briefly talk about at the end of the presentation. And finally, to strengthen the community. When we started, we had one initiative before BioExcel already in 2012 and 13 to bring together the computational biomolecular research communities because this is really fundamental for research and progress. So in our portfolio, we have several core applications. One of them is GROMOX, which was already presented. It's molecular dynamics simulation code. It's one of the most popular, maybe even the most popular. It's difficult to assess exactly the number of users, but there could be probably 10,000 users or more, like especially through the years, that are using it from academia and research. Primarily, it's used for modeling of proteins, DNA, cells, membranes, but it's also used for soft matter physics as well. LAMPS is one application that is very popular in soft matter physics, but GROMOX also is often used for polymers, for example, or droplets. I will go a bit quickly through the slides. What makes GROMOX rather specific and very useful for HPC is that throughout the history of the code, the main focus has been on portability and bottom-up performance oriented design, which has allowed the code to be the way it's written at the moment is that it's very easy to port it to any new architecture. Now, fine-tuning it for any architecture can require a lot of effort, but just getting it to run is much easier than with other codes. And GROMOX adopts a rather complex multi-level hierarchical parallelization. So that means that the code implements different methods and software stacks to ensure efficient parallelization within the node on an HPC system, such as OpenMP multi-threading, SIMD instruction layer, or GPUs if that's available. Then internode, like how do we ensure that every node in the cluster is communicating properly and efficiently? This is a completely different type of work that needs to be done. And finally, multi-level load balancing, having a dynamic and task balancing as the application progresses. This is not interesting only from software engineering perspective, but also from user perspective, that it makes it straightforward even for novice users to make the best out of any architecture. For those of you who are a bit more interested in the internals, there are a number of papers on how this is implemented in GROMOX, but basically what the kernels do in the original homogeneous scheme where you have only, let's say, CPUs, each type of calculations are done sequentially one after the other. Now, each of these boxes will be done in parallel, But until you're done, let's say with the bonded interactions, you don't compute the non-bonded and so forth. With the GPUs or other accelerators that came on the market in the past, the first one was to offload the green boxes, which typically take the most time, to the GPU, but this requires frequent communication with the CPU and eventually becomes the bottleneck. What is available in the later versions is it becomes a bit more complex, where more and more of the types of calculations, the purple, blue, orange boxes, can be offloaded also to the GPU. And now you can imagine that you have different HPC systems with one or two very powerful GPUs and just a few CPU cores on each node is one type of system. Another one, you may have a lot of CPU cores on the node and one slow GPU. So these scenarios affect the performance in a great way. And what Gromax is trying automatically to do usually is to optimize for that. But you as a user will be in charge of deciding how you want to schedule this, and you can fine tune it by yourself. The way the source code was redesigned, also data structures many years ago allow it for very easily porting it to new architectures, irrespective of the SIMD width of the parallel processors. We've been working very closely with pretty much with all the major hardware vendors, NVIDIA, Intel, AMD, with all of them on ensuring that the code is highly tuned for each of their processors. This has been great, and this type of code design work is the way to go for software applications. So if you have code and you're very interested to optimize it for one of these platforms, the companies do put a lot of effort into supporting researchers in the optimization. So if you're interested, you can outreach directly with them, or can get in touch with me and I can put you in touch with the developers. I will skip some of this a bit more detailed work. This is presenting how we did porting on with SQL on AMD systems and also using HIP. This is another software stack that is mainly used by AMD for their processors. Something very practical for those of you who are not software developers but mainly using Gromax in your research work is we did together with the CSC team at Lumi, which is at the moment the fastest supercomputer in Europe, testing of how in the best way you can use the system. So Lumi is a gigantic system with almost half, 375 petaflops, almost half exaflop performance, sustained computing performance, which is quite impressive. The machine has two different partitions. One partition is with CPUs only, and the other one is with fewer CPUs but 8 GPU processors on it. And what is very important for the users is when you send your job, like, do you want to send your job only on the CPU partition? Do you want to send it only on the GPU partition? And how do you, if you do it on the GPU, what's the best way to pack your simulation data onto these powerful nodes? So we did the following tests and benchmarks. We used a system of about 1 million atoms, which is rather large. It's not huge, but still a big system. And tested the performance on the CPU-only nodes and on those on the GPUs, which, as you can see from the bars, you get much better utilization using the GPUs. And that's because you have this very big system and you're able to fill in all the memory of the GPUs here, because they have a big memory. Gromox in general doesn't use that much memory. So to be most efficient, it's good to pack a lot of data there, which takes us to the second test, where on the GPU nodes, we try to see whether we can pack multiple simulations at the same time onto the same GPU. And here we have a much smaller system of about 100,000 atoms, which is very typical for biomolecular simulations, let's say, of membrane proteins or so weighted ones. And you can see how it's possible to pack, let's say, 4, in this case, is the optimal number onto the same card. So why is this important? Well, when you get your allocation on a system, you get node hours or CPU hours, and if you're not using it efficiently, you're just burning electricity, and you don't get good return on investment for your time. So if you get large allocations, I recommend that you spend a bit of time on fine tuning and finding out what's the optimal way to run your simulations. As a summary of Lumi, of the Lumi work, as mentioned, for large systems, it's possible to scale on multiple GPUs, and you can maximize the utilization of the GPUs by running multiple independent trajectories per one node. Another thing that is worth noting is that running on the GPU nodes, to have 100 microsecond simulation, you use only 2% of all of them, while if you're using only CPU nodes, then you need almost half of the partition. Gromo's development team is very big. The core of it is in Stockholm, in KTH, people in PDC and Science for Life Lab, but there's many other people who are contributing around and have been and continue to contribute. One very useful application of Gromox is that its ability to calculate free energies, and maybe some of you who are interested in computational drug design are already using that feature. Now it's possible to use Gromox only. However, in BioExcel, there's an application called PMX, which is kind of a wrapper around it, like a controller that uses Gromox on the backend, but automates all the intermediate steps that you need to do to calculate these free energy calculations. So it makes it much more user-friendly. As I mentioned earlier, user-friendliness of software is very critical for productive research work. So in a typical free energy calculation for drug screening, what one does is to calculate the delta G, is the difference in the free energy between having one ligand replaced with another ligand, or removing the ligand completely or changing. So this cycle of the free energy actually gives you at the end how much stronger the blue drug, for example, binds compared to the red one. And this you can do not only for drugs, but you can do it also with small peptides, if you want, or DNA or other segments. It doesn't really matter what the species are, as long as you have the proper force field parameters developed for it. And you can do it with binding to proteins or binding to DNA or any mixture of those. What is usually very difficult and very valuable in energy calculation is to be able to calculate the absolute protein ligand binding. And for that, you need to be able to calculate it with a very big precision. So quite some time ago, we did a very large simulation testing. We ran several hundreds of free energy estimations for a number of systems. And well, this type of calculation is inherently embarrassingly parallel, so you could very easily parallelize it over many nodes. What it showed is indeed in just a few days you can screen a very big library of ligands, and what you get as delta G. So was the difference in the difference of the change of the change of the free energy relative to different force fields. It's very consistent and very good. Since it's embarrassingly parallel, it fits very well to run it also on cloud infrastructure. So you don't need in some cases to have access to HPC. Depending on your type of problem, you can run this at scale on, say, on Amazon or Google or Microsoft, any type of cloud infrastructure. And it's the results you get with PMX and GROMAX are comparable. Sometimes excel those that you get with commercial, very expensive software. So we see a big uptake of the codes by pharma companies and not only academic researchers. The main developer team of PMX is in Göttingen in Germany, the Max Planck Institute, in the group of Bert de Groot. Now moving a bit away from particle simulations, molecular dynamics, to integrative modeling, another called HADOC, which is also very popular, also with many thousands of users worldwide for integrative modeling. So in docking, one tries to understand what's the most efficient shape of these molecular complexes, which could be between proteins, could be a protein and a ligand, or DNA, any type of species. What one would usually do when you try to say you have two molecules and you try to find how they bind, you need to explore the whole conformational landscape and to estimate for each of these conformations what's the binding energy, and this is extremely time consuming. What one can do to speed up considerably this search is when you have information driven search where you incorporate additional experimental or other simulations data that limits the search of this surface, and this funnel becomes much smaller. For example, you can incorporate data from cryo-electron microscopy, from bioinformatics studies. NMR often gives you a lot of useful information of proximity of atoms and shapes and so on. So a lot of this information can be integrated during the modeling process, and HADOC allows you to do that in a very efficient way. HADOC's been participating every year in the performance competitions such as Capri with excellent results. For example, some years ago, you can see that it was able to identify the target structure in the it was among the top ranked models, and this happens almost every year. We've also done large-scale modeling. This is a bit older data, but it still holds. We are doing more and more of that when trying to run it at very large scale, HADOC docking on multiple process with very good performance. Often for HADOC, it's more of an embarrassingly parallel problem, high throughput type of simulation. So it fits well also on cloud. and Hadoop is developed at University of Utrecht, the group of Alexander Bonven, and with many people contributing to that. Now, the last major application I'm going to present is BioExcel building blocks. It's an automation workflow. Those of you who are doing science with all of this software, you know that very rarely, I would say almost never, you use just one application to solve a specific problem. You need a number of codes to build complex workflows. And what hurts productivity of researchers is when you need to do a lot of pre-processing and post-processing of the results. Even if GROMACS runs 10 times faster, eventually you get limited by how quickly you can change your input files or decide whether the results you like them and you have to rerun again. So packing everything into a workflow is fundamental, and this is the way to go in future, especially when you're using very large HPC machines. And the number of tools that one uses in biomolecular simulations is just staggering. There's so many of them. So what we did in BioExcel is to build this workflow platform, BioBB, where similarly to, if you think of a metaphor for Lego building blocks, you can create your own workflows, combining different modules within the workflow manager. And this is rather awful. 2 years ago, but they were already at the time, there were almost 20 different types of modules for many of the popular problems that you recognize with GROMACS, AMBER, also for quantum mechanics, but PMX as well, of course. The number is growing all the time, and the group at IRB is very active. I will skip this one. We've shown it also with workflows. One can achieve great scalability here. You can see that running on multiple nodes, performance drops around 300, but still this is great. If you're able to just push a button, send your simulation to the supercomputer and forget about it. I will not talk about malleability. You can find a lot of information about it on the BioBB website, where there's a lot of tutorials and sample workflows that you can just pick up and use by yourself. There's also information how to write your own modules if you're interested in doing that. The developer team of Modesto Orozco's group in Institute for Research in Biomedicine, IRB in Barcelona, is very active, and they can help you if you're interested to try it out and introduce it into your work. Now, finally, a few words about the community aspect, because solving grand challenges requires not only very fast supercomputers, very efficient software and expertise, but also we need to make sure that the community is working closely together and they have the necessary skills to do that. At BioExcel, we provide support forums for all of the applications online. We have regular webinar series where we feature key codes in the field of biomolecular research, presentations of new tools or updates. A lot of documentation can be found on those.bioexcel.eu, and also the training program. One thing we started a couple of years ago is so-called ambassador program, where we are trying to spread the outreach of BioExcel across Europe. So our ambition is to have pretty much in every country in Europe a person, usually a senior PI, scientist, professor, assistant professor, who is a user of the application and can be a focal point for the local community. We have almost in all of the countries in Greece, our ambassador is Zoe Cornil, whom you probably know. And what we do with the ambassador is the following. We have defined 7 regions in Europe. And for each of these regions, we are running local workshops. Here's the table with what's the plan for the workshops in the different regions. How is that useful for you? Well, as you see, in 2026, we will have a workshop in Greece, Cyprus, and Turkey, the Aegean edition, so-called. The way it works is that the ambassadors in those countries will outreach to you, to the community, and find out what type of training you need the most. Let's say maybe you know very well how to run GROMAX, but most of the people would like to learn more how to use with PMX to do free energy and also to write custom workflows with BioBB. So the ambassadors, together with the local communities, define the content of the workshop, and BioExcel sends the trainers to the region to do a training, which is usually 2 or 3 days of hands-on. During the training, users usually have access to HPC infrastructure where they can run the simulations. As of now, we don't have a fixed time. Most likely will be in spring 26, or we don't know yet whether maybe this will be in Turkey, because it's easier for Turkish, or rather, it's difficult for Turkish researchers to go to Europe because they need visas. Only for that reason, we may do it in Turkey, but this will be discussed further with the ambassadors there. So if you are interested in that, I recommend you subscribe to our newsletter or follow the website when we announce the workshop. And you can also follow us on Twitter. Our YouTube channel has maybe now 80 webinars already, and yeah, there's a lot of useful information there. So that's all from me. Thank you for today, and I'm happy to answer any questions you may have.